Hi, welcome to my presentation. I should probably get dressed up for this. That's better. Brendan Nichols, aka Themightymiracleman here, presenting on the paper Particulate matter induces inflammatory cytokine production via activation of NFkB by TLR5-NOx4-ROS signaling in human keratinocyte and mouse skin. The question that they were seeking to answer is if particulate matter increased pro-inflammatory cytokines. Sung Rao Hong Kong and others wanted to see how things affected the keratinocytes. Here's some of the findings. Findings. So what is IL-6 or interleukin-6? Index is both a pro-inflammatory and anti-inflammatory cytokine. It was originally discovered as a B-cell differentiation factor that, quote, regulates the immune response, hemopoiesis, the acute phase response, and inflammation. IL-6 is produced by various types of cell and influences various cell types and has multiple biological activities through its unique receptor system. IL-6 exerts its biological activities through two molecules, IL-6R or IL-6 receptor and GP130, unquote. Mahara et al. 2012. That was a mouthful. I need more coffee. They talked about GP132, but what is that? That's relatively simple. G-glycoP protein is a glycoprotein that is a transmembrane protein and acts as a receptor for the cytokines. The results of this study found that ligation of TLR5 with the particulate matter labeled PM2.5 stimulated NOx4 to generate ROS or reactive oxygen species which in turn elicited the activation of NFkB and promotes secretion of pro-inflammatory cytokines, including IL-6. It did this by histone methylation and decreasing the binding affinity of the transcriptional repressor and instead inducing activation of transcriptional activator to the promoter. What this all means is that particulate matter seems to induce pro-inflammatory cytokines. This can be seen in the graph and the control compared to those mice treated with PM 2.5. Now on to TLRs. These recognize pathogen-associated molecular patterns, or PEP for short. Binding of PEP to TLRs induces inflammation. It is thought that by promoting TLRs, the excessive TLRs promote the pro-inflammatory genes. So I kind of skipped ahead to figure 6 because that one was the most understandable for what I was talking about. And now let's go back and look at some of the other figures. You can see here that when the cells were incubated with the particulate matter, instead of just a control, the cytokine concentration was much greater. I repeat, much greater, much greater, much greater. Here we see the TLR5 inducing increased cytokine production. What are all those blue dots for, you ask? Those are fluorescent probes of the DNA with the labeled RNA. It changes a protein from non-fluorescing to fluorescing. This graph shows some of the effects of interaction between TLR5 and NOx4. You can see that the effects of the interaction between those two create more reactive oxygen species, or ROS, which are redox signaling molecules. These redox signaling molecules act by inducing PEP pathogen-associated molecular patterns do cause inflammation by the binding of PEP to TLRs, and that leads to the inflammatory response. This is the paper I reported on an APA format. I hope you guys can find it and check it out. It's pretty cool. I love redox biology.